Hello everyone, today we'll be talking about endosymbiotic theory. Before we can get into endosymbiotic theory, what it is, why it is important, etc., there is a certain prerequisite. That is, you must know the difference between eukaryotic cells and prokaryotic cells. All types of cells are divided into two categories, essentially. That is, prokaryotes and eukaryotes. So, well, a eukaryote sort of looks like this, maybe. So yeah, that's basically what it looks like. Now, a prokaryote is it has a rod shape like that, and a eukaryote is free-form shaped. Now, what are the differences? Well, for one, eukaryotes are immensely larger. See, immensely larger, whilst these are much smaller. I'm not even gonna write. Never mind. Now, prokaryotes, there are a lot more eukaryotes than there are prokaryotes. So you should also know that. So now eukaryotes reproduce by what is called binary fission. Now this is a rod-shaped prokaryote that I'm drawing. It will become bigger and bigger until it is completely destroyed and another daughter cell is formed. Pretty much how it's done, an overly simplified version of how it's done. Now, eukaryotes produced by sexual reproduction, which, you know, mitosis, pretty similar. Understand the concept. Now, the biggest difference is that a prokaryotic cell does not have a membrane, meaning its insides are just floating around. It has a membrane, I'm sorry, but its organelles are not membrane bound. So if this is a prokaryote, inside its membranes are essentially just they're floating around. That's a poor representation, but you understand. Now, the inside of a eukaryotic cell has cytoplasm and has cytoplasm and a cytoskeleton, but we'll talk about that a little bit later. Let's see. Whoops. Choose a different shade. Oh, that's not working? Well, let's just pretend that this is this black area here is the cytoplasm. Now, inside of there are the numerous organelles. It's mitochondria. Now that we understand the basic difference between these two, you know, the rod-shaped... Oh, I'm sorry, this is not drawn to scale, by the way. These are actually quite smaller. So now that we understand the difference between these two, we can go into actual endosymbiotic theory. In the beginning, there was only one type of cell, and that is the prokaryote. Many of these prokaryotes were actually anaerobic. That means basically that they can't live in an oxygen environment. So there we go. No, it's good enough. Now, the theory explains why the eukaryote exists. And now, you know, the eukaryote is quite more complex than this rod-shaped prokaryote. Now we can see here, prokaryotes were all rod-shaped, but eukaryotes, which, like I said, are free-form shaped. This is essentially what they look like. They're free-form shaped. So eukaryotes, this is cytoplasm, by the way. Their freeform quality, basically, it really serves two functions. Now, if you don't have a cell wall, the metabolic processes, that what I mean is the decomposition of the food, not decomposition, the consumption of the food happens quite, it happens quicker. Now, the next thing is that it's easier for it to move around. This isn't everything inside of eukaryotic cell. I'll label them as I go around. Now, the next major thing we see is the cytoskeleton forming. Well, and for color's sake, I'll just make this a bit darker. Not saying that it actually is this color, but whatever. So, the cytoplasm extends all across. In eukaryotes, they have this cytoskeleton, and in prokaryotes, they do not. They most definitely do not. Now, the purpose of this light green area is to represent the cytoplasm, which inside of the cytoplasm are the numerous organelles, which are held down by the cytoplasm itself. So, this skeleton here also forms, it makes it easier for chemicals to be transported. Now, like I've said before, prokaryotes do not have a nucleus, they have a nucleoid. See this right here. Basically ends with a eukaryote, we do see the formation of a closed nuclear membrane. Let's pretend that's it. Now, inside of here there are little tiny chromosomes, but it's not closed like it is here, very neat and organized, and sort of the brain of the cell. So now that we have the shape down, let's talk about the actual organelles. All eukaryotes, that is these are the eukaryotes. All of these were originally anaerobic, but then came along these things called peroxisomes. Let's say that this is what a peroxisome looks like. Many different little bridges. Now, this is basically a peroxisome is basically aerobic, which means that it can last in an oxygen-rich environment. So this eukaryote, which is currently anaerobic, will now take this peroxisome and consume it, like literally consume it. And now the eukaryote now has the ability to thrive in an oxygen-rich environment. And really, that is basically what endosymbiotic theory is. It is the taking in of other organisms, living with them in a mutually beneficial sort of environment. And then, as time went on, endosymbiotic theory came to an end, and the DNA of these peroxisomes became permanently entwined with the cell itself. So now we have the DNA of the peroxisome now stored in the nucleus, so the cell will now forever remember to also make this structure, which is the peroxisome. Now that's the core of endosymbiotic 3, and I hope that you learned something from this.